Dear students, in this unit we will talk about the chromosomal mutations. Now, we have studied in the previous lectures that point mutation or the mutations are a very important part in the course of evolution, in which a single nucleotide of the DNA is changed and it has significant consequences. But in this unit we will talk about mutation at a larger level, at the chromosomal mutation. So a mutation which involves a long segment of DNA, a significant portion of a chromosome, that mutation is known as chromosomal mutation. These mutation can involve either deletion of a significant portion of the DNA, hence deletion of genes which are present at that specific locations. It could either be insertion of another portion of a chromosome in another chromosome, which can result in the halting or in disruption of the function of that typical gene where this chromosomal part has been inserted, or it could be inversion of the section of DNA. This section of DNA can be inverted, so the sequence is changed, and this change of sequence ultimately result in the change of the nucleotides and change in the structure of the proteins, which is very dangerous, and in many genetic diseases it is very evident. Now, in some cases, these deleted section of that chromosome may attach to other chromosome, and it becomes the addition in that gamete or that individual. It also disrupt both the chromosome that loses the DNA and that chromosome which gained it. So it disrupts the function of both chromosomes. There are 4 different types of chromosomal mutations: deletion, then we have translocation, then we have duplication, and inversions. Deletion means any chromosome mutation resulting in a significant loss of genetic material is known as deletion mutation, and it is mostly very lethal for an organism. Then we have some other mutation as well, and these mutation at chromosomal level, they can be easily detected by either microscopic examination or either by genetic analysis or by both. Before the advent of molecular biology, normally the chromosomal mutations used to be detected using microscope, and there is a specific technique known as karyotyping. Karyotyping was used to specifically determine the normality or abnormality of the chromosomes, while in genetic analysis now sequencing, next generation sequencing, and many other techniques are available which can combinedly being used in order to study the chromosomal mutations. This distinguishes chromosomal mutations from gene mutations. Gene mutation cannot be detected using microscope because it is a very minute at a very simple nucleotide level where microscope is unable to identify. Now, the example of translocation is evident here. You can see this is normal chromosome number 9, and we have another chromosome number 22. They are present in a normal individual, but somehow during the process of cell division, crossing over or fertilization, here in this case, in the crossing over, as you can see that this chromosome size has decreased significantly as compared to a normal chromosome, which is evident from here that ABL gene is normally present at chromosome number 9, but the BCR gene is normally present at chromosomal number 22, but their crossing over has resulted in an abnormal chromosome which has both these genes at a single small chromosome, which is known as Philadelphia chromosome. This chromosomal mutation results in a very deleterious effects. These mutations are result of certain accidents or irregularities in chromosome at the time of cell division, crossing over, or fertilization. These incidents cause alteration in the morphology and number of chromosome, which ultimately leads towards various genetic diseases. Now, these mutations lead to abnormalities in the function of cell as well as organisms, as these chromosomal mutation can result in abnormal gene number or positions. These are known to cause different genetic diseases that can be hereditary and are transferred from one generation to another generation. And this often is the result. This results in the very serious consequences for the individual which harbor those chromosomal mutations.